Hello everyone. Today we will study what is incomplete dominance. In the previous video lecture, we saw a cross in pea plant considering character like plant height. And we saw that in the F1 generation plants, the plants were genotypically hybrid or heterozygous, that is capital T small t, but the height of the plant was still same tall as the pure tall parent with capital T genotype. That means the capital T, the tallness was completely dominant over the short height in the garden pea plants. And that simply explains the law of dominance. That is, in hybrid condition, only one of the character is completely expressed and the other one is suppressed. So this is simply the law of dominance. Now the question is the law of dominance applicable to all the genes? Like, is this law universal? Then the answer is no. There are some genes which have a slightly different pattern of inheritance. So one such exception which we will see today is incomplete dominance. In case of incomplete dominance, we see that neither of the alleles is dominant over the other. So if we take example like if we have a gene with two alternative forms, capital R and small r, then according to law of dominance, only one copy like the capital R should have been completely dominant. But in this case, that is of incomplete dominance, we see that neither of the alleles is dominating, and hence in case of hybrid condition, that is genotype capital R small r, we get intermediate phenotype between the two parental characters. To study this, we take an example, that is flower color inheritance in Antirrhinum majus, that is snapdragon plant or simply known as dog flower plant. Same example of flower color inheritance can be applied to Mirabilis jalapa, that is 4 o'clock plant. Let's see the cross. Now the basic steps in the cross are same as we studied in the garden pea plant. So first we start with the parental generation. The parent should be pure and contrasting. So as you can see, one plant has the red color flowers and the other one has white color flowers. And here, first thing which was done is both the plants are obtained in pure condition. So, red color, here the gene, the allelic form which controls red color, right? Same copy is repeated here. So, capital R, capital R. And in white color, we see small r, small r. So, pure plants. Now, first step will be to form gametes. So, both these plants will form the gametes as this first plant has the genotype capital R, and during gamete formation, the chromosomes separate and hence the genes, the alleles also separate. So this capital R and this second capital R will also separate. But this will not create any difference, right? We will have only one type of gamete, that is all the gametes will contain a copy of capital R. Similarly, for the second plant with white color flowers, we see the genotype is homozygous, right? Small r. So here also when the small r will separate, we will get only one type of gamete, that is containing one copy of small r. Okay, now when both these gametes will fuse, we will get the first filial generation, right? First offsprings generation. And here, as we can see directly, if capital R and small r containing gametes fuse, the offsprings, the F1 generation offsprings will have the genotype capital R small r. But what's different over here? The character of F1 generation is not matching with any of the parent, but it is an intermediate character. So we see here pink color flowers are obtained, and this itself tells that this character has incomplete dominance, right? In hybrid condition, we see an intermediate character. Now, what will be the next step? Same as the normal monohybrid cross. This is also a monohybrid cross, but here the law of dominance is not applicable. Here we have incomplete dominance. Okay, so the same step will be there. We will do self-pollination in the F1 generation plants, and we will have the female and the male gametes. As the genotype of F1 generation plants is capital R small r, this capital R and small r will separate, and hence 50% of the gametes, let's say the female gametes, 50% of the female gametes will have capital R and the remaining 50% will have small r. Similarly, in male also 50% with capital R and 50% with small r. So here two types of male and female gametes are produced as the plant was hybrid. Now, the female and the male gametes will fuse. So all the possible combinations of offsprings are shown in this Punnett square. So if capital R and this capital R fuse, right, these two gametes, then we will get the offspring with genotype capital R, and as we know, both the copies are same and capital R codes for red color. We see the plant produced here will have red color flowers. Now, if this small r and this capital R gamete fuse, we see the genotype of the offspring is capital R, small r, and as this is hybrid, and in this example, incomplete dominance is observed. So here the plant will have pink color flowers. Similarly, here this plant will have capital R, so pink color flowers. And now the last, fourth plant here, small r gametes are fused. So if we have the genotype small r, we will get white color flowers. So what we see in the F2 generation is that out of 4 plants, we have 1 plant with red color flowers, 2 plants with pink color flowers, and 1 with white color flowers. This is very different from what we saw in the previous lecture when we saw crosses of the pea plant, wherein if we took the tall and short plant, made a cross, then in F2 generation we saw that we get 3 tall and 1 short. So the phenotypic ratio there was 3:1, but in this case we have 3 different phenotypes, 3 different physical expressions, like color is the phenotype here. So 1 red, 2 pink and 1 white. So 1:2:1 will be the phenotypic ratio, and the genotypic ratio will be same as the previous cross, as we have 1 capital R, 2 plants of capital R, and 1 of small r. So what we can say here is the phenotypic and genotypic ratio of F2 generation plants in case of incomplete dominance, both the ratios are same. 1:2:1. Here also you can see genotypic ratio is 1:2:1. So this is also very important character in case of incomplete dominance. Both phenotypic and genotypic ratios are same. As the hybrid shows intermediate character, that is why we use the term incomplete dominance. Also, make sure that you understand here that though these F1 plants had pink color flowers, right, and genotype is capital R, small r, when the gametes formed, right, capital R and small r separated as such, right, there was no like blending between the alleles. So law of segregation is still applicable, but law of dominance is not followed here, as the hybrid is showing an intermediate character. So this itself explains what is incomplete dominance. Now, let's take one more example again, which is given in your NCERT textbook, that is inheritance of seed shape and starch grain size in pea plants. This is in the normal garden pea plant. Okay. Now, Mendel studied the seed shape character also, right? We know that in case of peas, we have 2 seed shapes, alternative forms, round and wrinkled. So, let's simply say that the G, usually we write it as R because round is dominant, but we will use the same letter here as used in your textbook with respect to this example. So, if we take the character of seed shape and make the cross, what we can see? First, let's say we take the plant with capital B, a pure plant with round shape, okay, and cross it with the contrasting form, again pure, small b. This has wrinkled shape seeds. Now, when gametes will form, what we will see? This capital B will separate, and we will get only one type of gamete, which is capital B. And in this second plant with wrinkled seeds, small b will separate. Whether you take the first one or second one, you have only one type of gamete, that is small b. Now, these two gametes will fuse and you will get the offspring capital B, small b. Now, in this case, we see that the genotype is hybrid, heterozygous, but the phenotype, what phenotype do we observe? Complete round shape as seen in this first parent. Okay, round shape as seen in this first parent. That means here capital B character, okay, the round shaped character is completely dominant over the wrinkled, that is the small b allele. So this explains complete dominance. This explains complete dominance, as the normal other characters also we see in the pea plants, okay, like the plant height. But the same gene, okay, same gene B. Let's take the same plant, okay, and make the cross. We have the same gene B. If the genotype is capital B, we see that in the seeds, the storage material, starch, okay, the starch grain size here is different. So in case of capital B, we see that the starch grain size is large, okay, large grains of the the starch, large grains of starch, okay. And in small b, we have small size, small grains of starch. Okay. Now we make the cross. Gamete formation will be the same. Capital B will separate. So we get only one type of gamete. Small b will separate. So again we get one type of gamete only. Now what we can see here, both these gametes will fuse and you will get capital B, small b. What we expect according to complete dominance is the grain size, starch grain size should be matching with one of the parental characters, like let's say the large size. But it is not so. If we take this hybrid plant, capital B, small b, we see that the starch grain size is intermediate, medium size. Starch grains are intermediate, okay, medium in size. So what do we understand from this example? Okay, same gene B. If we look at the character seed shape, it is showing complete dominance. But the same gene and coding for a different character, okay, that is starch grain size, and in hybrid condition it is showing intermediate starch grains. As we can see, there is an intermediate character. Actually this is incomplete dominance. What is this? Incomplete dominance. Okay, question is same gene, but in one case there is complete dominance and in the second case we have incomplete dominance. So what we can conclude here is actually dominance is not an autonomous feature of the gene itself. Okay, the gene sequence, like you will have actually some sequence of nucleotides in the specific gene, capital B and small b. That sequence, that gene is actually not like dominating, but we decide which character is dominant, which is recessive based on the character expressed. So we look for the phenotype. Now in this case, the hybrid plant have round shaped seeds, and that is why we say that round shape is dominant. And now which allele controls round? Capital B. So we simply refer it to as dominant allele, but actually the character is dominant. Okay. So here we get introduced to one more term, that is pleiotropy. You can see the paragraph from your textbook itself, wherein the same cross is given. You have the gene B. It has two alternative forms, two alleles, capital B, small b. If we consider seed shape, then we see that in hybrid condition we have round shape. But if you take the starch grain size, then in hybrid condition, capital B, small b, we have intermediate size of the starch grain. So this single gene is controlling two different characters. That means more than one effect. This phenomenon, see the first line. First line, occasionally a gene product may produce more than one effect, right? One gene. See, gene is one only, it has two different forms. Capital B and small b are different alleles, but the gene is same, right? So this same gene, its product is actually producing more than one effect, right? It is controlling the seed shape as well as the starch grain size. This phenomenon is called as pleiotropy. What is this called? Pleiotropy. One gene controls many characters. Okay, and in using the same example, we can see that though we have the same gene, in one case like seed shape, it is expressing as complete dominance, and in case of starch grain size it is showing incomplete dominance. So what we can conclude, see here in this paragraph, right, very important, see, dominance is not an autonomous feature of a gene or the product that it has information for. Then what does dominance depend upon? It depends as much as on the gene product and the production of a particular phenotype from the product. So this is very important. Dominance is not an autonomous feature of the gene, that sequence, or simply the product, but along with the product, the phenotype expressed is very important. We look for the phenotype, a particular phenotype, and then we decide whether it is complete dominance or incomplete dominance. For example, we saw that the hybrid plants in Mirabilis jalapa or Antirrhinum produced pink color flowers, and based on this pink color character, we explain that this is incomplete dominance, right? Simply by saying capital R, small r in the flower color inheritance cross of Antirrhinum majus, we cannot say that it is incomplete dominance. But by looking at the pink color flowers, we can say, okay, this is an intermediate character, so this is incomplete dominance. So what is important is product and the particular phenotype expressed, right? This is the deciding factor. And that is why we say dominance is not an autonomous feature of the gene itself. So I hope this video lecture was helpful for you. Today we saw what is incomplete dominance, then the cross using flower color inheritance in Antirrhinum majus or Mirabilis jalapa. So these were the examples for incomplete dominance. Plus starch grain size inheritance in pea plants is also an additional example for you in your syllabus for incomplete dominance. Okay, this is given here. It is not directly given in the main topic, but this is definitely the starch grain size is example of incomplete dominance. Also, in the same example, we see one gene is controlling two different characters, seed shape and the starch grain size inheritance, and as one gene is controlling more than one phenotype, this gene is a pleiotropic gene and the phenomenon is called pleiotropy. Okay, I hope this video lecture was helpful for you. Thank you.